The three stages of cell signaling are reception, transduction, and the response. Now, the first one is reception, where you will find that a signaling molecule is received by another molecule which is present on the plasma membrane. So generally, this molecule is known as receptor. This is maximum time this is a protein molecule, and we are going to see what are the different kinds of receptors present on the plasma membrane. What are the different roles of this kind of receptor? So in general, the receptor receives the signaling molecule, and as a result, the conformation of this receptor molecule, as these are all dynamic in nature, right? These are not fixed. Some protein molecules, when they are in action, so they make change their conformation, one form to another form. So here also you will observe that due to the interaction between this signaling molecule and this receptor molecule, some conformational change is happening inside the receptor molecules. And as a result, the other molecules inside that cell are activated. The next step is known as signal transduction. So this signaling molecule is not directly going inside the cell, but this receptor molecule is relaying the signal to the other molecules. So these relay molecules in a signal transduction pathway are ultimately generating a response, and which is basically including the activation of different cellular activity. Now these are the three steps of cell signaling pathway in general: reception, transduction, and response. Now think logically, when we are talking about cell signaling, so this is a case of uh, switch, right? Now in this example, you can see that the switch is on, isn't it? The signal is coming, the signal is moving towards this cell, and whatever activities, say for example, um, this signal is for some growth. Now the mRNA for that growth responsive proteins will be generated inside the cell, and the protein will be formed from that mRNA, and ultimately the cell will grow. But think logically, how far that cell will grow, how long this process will go on. If it is going on and on, obviously the cell will burst out, isn't it? So this switch must be off after some time. And now don't ask me that who is doing this uh, regulation. So that is also active research area. Now, how this kind of signaling switch is on, or when that will be inactivated, that depends on the cellular activity. So there is a feedback mechanism. When there is no need of that molecule, that pathway will be stopped. Okay, so this is a basic mechanism of cell signaling, that whenever a switch is on, that switch must be off after some time, and then again that switch can be on for the same purpose. Okay, so we have already talked about this, that signaling molecule binds to a receptor protein, causing it to change that state. This binding between the signal molecule, which is known as ligand, and the receptor is highly specific. And this specificity actually very much important for biological activities. And I told about the drug designing purpose also, this binding is very much important. Okay. The safe change in a receptor is often the initial transduction of the signal. Most signal receptors are plasma membrane proteins. Now we are going to check this kind of membrane proteins which are acting as receptor. So mostly three types of membrane receptors are found. One is G protein coupled receptors, another is receptor tyrosine kinases, and the third one is ion channel receptor. Now, most water soluble signal molecules bind to specific sites on the receptor protein that span the plasma membrane. So, in bioinformatics also, you will see in future that this kind of ligand and receptor binding are modeled via the computational tools and techniques, so that you can easily identify that how that ligand can bind to a particular receptor, uh, or whether the interaction is uh, very much specific or not, whether that kind of interaction can generate some other activities inside the cell or not. That kind of prediction we can do using bioinformatics tools and techniques. Now we are focusing on G protein coupled receptors. In short, it is known as GPCRs. These are the largest family of cell surface receptors. This protein is a plasma membrane receptor that works with the help of another protein, sorry, which is known as G protein. And you can easily understand that the name suggests that G protein coupled receptor. So it is a receptor which is acting with the help of another protein, which is actually the G protein. So that's why the name is G protein coupled receptor. G protein acts as an off on switch. Now we will see the mechanism of this. In principle, if the GDP is bound to that G protein, now you know that what is ATP, ADP, GTP, and GDP. So guanosine diphosphate, not triphosphate. Guanosine diphosphate is bound to that G protein. The G protein is inactive. So this is the principle. Okay, and when it is GTP bound, so then that protein is active. So this is the cartoon of G protein coupled receptor. You can see that 1, 2, 3, 4, 5, 6, 7. 7 transmembrane regions are there. So this is a huge complex. You can see it is a protein which has 7 transmembrane domain, and these are the loop regions. So this is a very beautiful structure. Although this is looking like a flat structure, actually it is forming like a channel. Okay, this is open form. So when it is in closed form, so it will look like this. It's a channel kind of thing. Okay, and 7. So you can see that my 5 fingers are there, but you can add 2 more fingers. So actually 7 such fingers are there on this plasma membrane. Okay, they are present on the plasma membrane, this region, and some regions are outside the cell and some regions remains inside the cell. Now this region which are found inside the cell, that can interact with the G protein. So G proteins are inside the cell like this. Okay, so this is a G protein, and as we know that when it is GDP bound, so then it is inactive. And here is a G protein coupled receptor present on the plasma membrane. Now, the ligand molecule or signaling molecule is bound to this GPCR, and a conformational change happens here inside this GPCR molecule. So the GDP bound inactive G protein can come and join to this region, and one GTP replaces that GDP, and thus the receptor is activated. Okay, and then what will happen? The GTP bound G protein, which is active now, that can relay the message to another molecule, another target molecule, which in turn will produce some cellular responses. And you know, the ligand can go out from the GPCR itself when the GTP bound activated G protein is away from its from this GPCR. And ultimately, what will happen when the cell response is no longer required? So then again, the G protein will be inactivated, and GTP will be hydrolyzed to form GDP and inorganic phosphate. So that's how the G protein is regulating the cell signaling mechanism. So next, we are going to see the dynamic features of this G protein coupled receptor. Just wait a second.
Coupled receptors have a large extracellular.
Grammarly?
Many G protein coupled. Many G protein coupled receptors have a large extracellular ligand binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the 2 activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G protein with a beta-gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G proteins. Upon prolonged stimulation, however, the receptors eventually inactivate, even if their activating ligands remain bound. In this case, a receptor kinase phosphorylates the cytosolic portions of the activated receptor. Once a receptor has been phosphorylated in this way, it binds with high affinity to an arrestin protein, which inactivates the receptor by preventing its interaction with G proteins. Arrestins also act as adapter proteins and recruit the phosphorylated receptors to clathrin-coated pits, from where the receptors are endocytosed. And afterwards, they can either be degraded in lysosomes or activate new signaling pathways.
So you can see the dynamic behavior of these G protein coupled receptors through this video link. This is given in this slide also. Next, another type of receptor molecule is known as receptor tyrosine kinase, in short RTK. These are also present on the membrane itself that attach phosphates to tyrosine. Now, a receptor tyrosine kinase can trigger multiple signal transduction pathway at once. It's very interesting. This can trigger multiple pathways within a short time period. And abnormal functioning of these molecules is associated with many types of cancers. This is very interesting in that point also. Now you can see in this diagram, the top one, that is number 1, the tyrosine kinase, receptor tyrosine kinase looks like this. It has also 3 sites. One site which is present outside the cell, one domain, another one is transmembrane region, and one region which remains inside the cytoplasm. So this is one molecule of RTK, receptor tyrosine kinase, and another molecule of tyrosine kinase is also found in the vicinity. That means in a close proximity, 2 molecules remain. And the cytoplasmic region contains some pockets where the tyrosine, which is an amino acid, right? So tyrosine, 3 tyrosine in each peptide, in each protein are present, and in this condition that is inactive. So these are known as inactive monomers. Okay, without the ligand. Now, when the ligand molecule comes and bind to both of these monomers, inactive monomers, they come closer and they join to form a dimer. Okay, so this is known as dimerization. Now, after that, some ATP molecules are hydrolyzed to form ADP, and as a result, this tyrosine group, this tyrosine amino acids, can be phosphorylated. So, unphosphorylated dimers now become phosphorylated dimer. Okay, and as a result of that, this phosphorylated regions of each monomer of this dimer molecule can generate some other signal towards the other protein. So you can see that in this diagram, some proteins can come which has some specific domain and which can interact to this phosphorylated tyrosine molecule, and thus this unphosphorylated molecule can be phosphorylated, and that will be activated, and which in turn can generate some cellular responses. Okay, so in this way, the receptor tyrosine kinases can help in the signal transduction pathway. Now we are going to see uh, the dynamic features of this molecule. Just wait a second.
Hello and welcome to today's. I will explain what these RTKs are and explain how they function. Receptor tyrosine kinases are transmembrane proteins which consist of a cytosolic part, and this one has tyrosine kinase activity. Receptor tyrosine kinases are involved in multiple signaling pathways in the cell. They phosphorylate target proteins which then induce intracellular signal transduction. This can then, in a downstream cascade, lead to, for example, gene regulation. Here we have a monomer of our receptor tyrosine kinase. There is a middle part which is embedded in the plasma membrane of the cell. The kinase also has an extracellular part here, and there is the intracellular part, which contains the tyrosine. The extracellular part over here is for the ligand to bind. So a growth factor, for example, can bind there, and this is called the ligand binding site. Inside the cell, facing in the cytosol, there is the part with the kinase activity, containing our tyrosines. So what are kinases? A kinase catalyzes the transfer of phosphate groups. So look at the kinase in blue. It can take away a phosphate from the trinucleotide and transfers that one to another protein. So it can phosphorylate a different protein. This protein, now phosphorylated, can then have downstream functions in the cell. So remember, a kinase is in general something which catalyzes the transfer of a phosphate group. In this case, we have a tyrosine kinase. Most receptor tyrosine kinases are available as monomers first. And in this state, the tyrosine residues are unphosphorylated. Now, once a ligand binds to the receptor site, and this occurs for both monomers, a process called dimerization is taking place. So both monomers change their conformation and a dimer is formed. And this is important because one receptor is phosphorylating the other one. After dimer formation, our tyrosine kinase is activated. And what happens here? We call this transphosphorylation, where one of these kinases phosphorylates the tyrosine of the other one here. After a while, all the tyrosine residues are phosphorylated. So now remember what our kinase is doing. They can phosphorylate different proteins. So we have a protein here in green, and this one can bind to the phosphate residue on the tyrosine. These proteins who are capable of binding to the phosphate have a SH2 domain. Another protein, illustrated in red, binds to the phosphate on the tyrosine here. The receptor tyrosine kinases are able to phosphorylate many different proteins. After a while, lots of different proteins can get phosphorylated by that. For illustration purposes, here we have the green protein, which is now in a phosphorylated state. This can be the start of several different signaling pathways. This protein in green, for example, can phosphorylate another protein downstream, and this in the end could, for example, lead to gene regulations. However, there are plenty of different intracellular signaling pathways which depend on receptor tyrosine kinases. I'm at the end of this talk. I hope
So far we have discussed about the activities of GPCR and receptor protein tyrosine. Now there is another channel protein which is also found on the plasma membrane, and that is known as ligand gated ion channel. This acts as a gate when the receptor changes its shape. When a signal molecule binds as a ligand to the receptor, the gate allows some specific ions, for example, sodium or calcium, through a channel in the receptor. Now you can see very simple mechanism. So this is a ligand gated ion channel which present on the plasma membrane. Now gate is closed, but when the signal molecule is present and it binds to the receptor molecule, the gate opens and the ions pass through this channel, and then that can generate some cellular response. When the ligand is going back, then the gate is closed and the channel remains closed in that process. So that's all for these three kinds of receptor molecules.